All right. Anyway, oh, I know where it was. This idea of this endomembrane system. Now remember, eukaryotes, Gael told us, membrane-bound organelles. And all of these organelles that you're seeing here are membrane-bound. We think that they all came about in the same origin some 2 million years ago, right? This was the folding of the cell membrane. If you look at all the membranes of all these organelles, they all have the exact same components. And that component is a song, which you will all sing for me later. But right now, I will sing it for you. It's just protein, phosphate, lipid, phosphate, protein. I didn't say it was a good song, but it's a song, right? So we think that at some point, these membranes folded in on themselves. They created the nuclear membrane, they created the ER, they created the vacuoles, the vesicles. All these things came from the exact same cell membrane. The other thing we know is that they're all similar in function. They all are connected to those proteins. What I mean by that is they're all going to be able to make that protein, starting with the nucleus, going to the ribosome, and then eventually being released out the cell membrane. These are the structures. These are the structures that what we call the endomembrane system. They're the nuclear envelope, the Golgi apparatus, the vacuoles, the ER, lysosomes, and the plasma membrane. They're not only connected by function, but they're connected by relatedness. They're all related to the same structure, and that is the plasma membrane. So in that nuclear envelope, we're going to have DNA. As you told me before, DNA is going to code for the order of amino acids. They're going to travel to that ER. We said that the ER had ribosomes on it, the rough ER anyway. And those ribosomes are where protein is going to be made. They're going to be secreted out of these Golgi apparatus. You don't need to write all these down. I'm just trying to get you to see the connection here. And those Golgi apparatus may produce a vacuole for storage, or they may produce lysosomes, a digestive enzyme. We talked about enzymes earlier. And they may be secreted out of that plasma membrane by a process called exocytosis. All of these membranes are connected. In a few minutes, I'm going to show you a couple cell structures that are not connected by this endomembrane system. We think they got there in a whole different way. So the first part of this endomembrane system that you need to know is the nucleus, right? And the nucleus has a nuclear envelope. It is semi-permeable. On your lab that is due today, don't forget to turn that lab in, please. If you need help, ask. But that membrane is semi-permeable. It's going to allow things out of the nucleus like RNA, but not allow DNA to get out, right? RNA is going to be the messenger that's going to travel to those ribosomes to put those amino acids in the proper order. The nuclear membrane is part of that endomembrane system. We think that it once folded from the original plasma membrane. Ari, did you get a question yet?
No.
I'm going to give you a question, okay?
Okay.
I warned you ahead of time. So inside that nucleus, we got this chromatin, and those chromatin become chromosomes, right? Chromosomes, do you know what shape those would be? Maybe you don't know that answer, but I'll throw that out there. Have you ever seen a chromosome? Any idea what the shapes are?
Which is shaped like an X.
Yeah, they're shaped like X's, right? We've seen those before. How many chromosomes we got? Nobody ever gets this one right, so I always ask this question to all my classes. Anybody know?
48 or something.
What's that?
48.
48's close. That's what a chimpanzee has. And I'll explain why the chimpanzee has a different chromosome number than us. You're close, though. Any other guesses?
We have 24 pairs.
Closer. That would give us 48, though.
26 pairs.
I don't know why I find this so amusing, but this is always the case. And it really doesn't.
32.
It really doesn't matter. It's just for my own amusement. But everybody gets it wrong. It's 46 chromosomes, 23 pairs. Okay.
Wait, so the XY chromosome is already,
Oh,
What's that?
so the XY is already included?
Yeah, they are included in that. Yeah.
Oh,
So
okay.
there's what we call 22 pair autosomal, and then the X and Y determine male and female. By the way, do you know how to tell a boy chromosome from a girl chromosome?
Smaller.
You look in their genes. Ah. Okay.
ah,
Everybody
I
laugh.
get it. Yep.
That was a
Oh,
bad
boy.
joke. Don't write it down. You look in their genes, get it? And also in the nucleus, you can see what they call nucleolus. These are little balls of RNA molecules, and they're going to make the ribosomes. Right? Remember, ribosomes are not membrane-bound organelles. They are just hunks of RNA. So bacteria can have those. As Gael pointed out, bacteria cannot have membrane-bound organelles, but they can have ribosomes. Is everybody okay? So here we're seeing a close-up of that endomembrane system. And in the bottom picture, again, if this pandemic ever clears up, you guys want to go over to Abbott and look at the electron microscopes. You know, right now I don't think they'll let us, but eventually they will. Anyway, you see the rough ER. And what that rough ER are ribosomes. They look rough under the microscope. And when I ask you on the test, which I will, what is the job of the ribosome, rough ER, you're going to tell me that they have ribosomes and their job is for protein synthesis. They make proteins. Questions on that?
Sorry, could you repeat that? Like, they're what and made for protein
Sure.
synthesis?
They're called rough ER because under the microscope, they look rough. They got ribosomes on them. And the job is always with ribosomes is that they are protein synthesis.
Okay.
They
So
make
they're
protein.
found in the ER.
This is where the primary structure of the protein comes together. Yeah, on the ER. Smooth ER is not. Now, these are bound ribosomes. So the memory cue, remember, is that they're bound to leave the cell, these particular ribosomes. I don't like this chapter much, A, because we can't look at these under the microscope, and B, because we're not going to memorize all these cell parts, but there are a couple important parts. I need you to know ribosomes. We're going to spend literally a whole chapter on one of these little dots down here. Oh, okay. That explains it. Thanks for whoever told me to look on the chat. That was right. All right, so here we go. We're going to see a blast from the past here. We're going to struggle a little bit. You guys feeling okay? All I see is blank faces. Well, actually, I'm seeing blank boxes. The rough ER is where proteins are going to be made. So the rough ER is the order of amino acids. What structure of protein we talking? Primary, secondary, tertiary. What do we got?
Primary.
Primary. What kind of bonds? Good job, Ari.
Peptide.
Peptide bonds, right? Ribosomes are where proteins are going to be made. Somebody translate this next sentence. Polypeptides are threaded into the sternal space of the ER. What's going on there? In simple terms. Any idea?
Let's see, Rosen polypeptides threaded in persistent space.
You're not talking to me yet?
They're converted to.
What do we got to do next after primary structure?
They're converted to secondary structures.
Yeah, they're going to go into a secondary structure. We're going to do that inside that ER. These proteins are going to fold into these native conformational shapes. They're going to become 3D. Why not do it out in the cytoplasm? Because those R groups are going to react with the wrong types of friends, right? Maybe your mother's told you that. Don't hang out with the wrong kinds of friends. If they stay in the cytoplasm, those R groups are going to react with everything in the cytoplasm. We want them inside that ER so that that's where they're going to fold. Simplified drawing would look something like this. Here's that ER. Here's the folds. Oh, can you see that at all? They're just going to become three-dimensionally shaped. Now, gifted student status, extra credit. The next sentence is going to take a little bit to translate. It says oligosaccharides bonds to the protein. What does that mean, oligosaccharides? You've never heard that term. And here's what I'm talking about in terms of internalizing and not memorizing. We are all going to do well on this exam because we're going to eventually all figure this out. What do I mean by oligosaccharides? You never heard the term, but what would you guess? Throw something out there and don't be afraid to be wrong.
Polysaccharide.
Okay, you heard of polysaccharides. So what would you guess an oligosaccharide to be? Take a guess. Is it a sugar? Is it a protein? What do you think?
Some type of sugar.
Big sugar.
Some type of sugar. You see what I'm talking about? You're going to know enough biology, not only for this exam, but for college, if you're buying into what I'm talking about, right? We just saw the word saccharides and we're thinking, yeah, it's a sugar. Now, this is not a real big sugar. Jada showed us that big cellulose sugar. This is a smaller sugar. It's bigger than a disaccharide, but it's smaller than a polysaccharide. Make sense?
So
Yeah, go ahead.
yeah, so I don't know. I just came up with a little strategy, I guess, like of how to internalize.
Please.
Like, yeah, for me, like I guess when you see the word oligosaccharide, you just disregard the saccharide since like you know it's already a sugar, so you just really need to look at the oligio.
That's what I'm talking about, right? Unless we become intimidated, we'll be able to answer all these questions, or most of them, including Thursday. And again, Thursday, I'm really want to see what we can do. I got real questions from a real AP exam. They're all fair game because they're all about stuff we talked about. And I want you to be able to try to tie those together. Again, what kind of percentage we talking for a successful AP score? We're looking at 100%, Denise? No.
75%.
Yeah, 75 would be awesome. It's a given. We're going on that 5 wall that I'm making, right? And we need about 70%. So don't be intimidated, okay? Finally, that protein is going to have to leave the cell, and it's going to leave through the cell membrane. It's no big deal because if you'll recall, the vesicle that we're going to put it in, this little vacuole, if you will, is going to be the same component of the cell membrane anyway. And so it's just going to fuse with the cell and let that protein out. Anybody know about diabetes in here? What protein do you got to produce that diabetics do not, at least in type 1? What protein has to be released out of the cell?
Is that insulin?
Insulin, right? So this could be the whole roadway to insulin. In my pancreas, on my rough ER, I got these proteins. I'm putting them together by primary structure. They're going to fold to the right shape inside the ER. They're going to fuse with a vesicle and be released into my blood. At least I hope every time I eat, right? That sugar is going to be absorbed by the insulin. Not much fun today. I know. I apologize. Now, there is smooth ER. Somebody get this question right, please. Even if I never mentioned smooth ER, if they asked you on the test, if rough ER has ribosomes, why do they call it smooth ER?
No ribosomes.
No ribosomes, right? That's all there is to it. And these guys do not make protein. They're going to do things like synthesize lipids. They're going to change glycogen into glucose. That should mean something to us, glycogen into glucose. Remember, glycogen is how we stored our sugar. We ate it as starch, and then we just convert it into glycogen. It's the same hexagons. It detoxifies drugs and poisons, and it stores calcium. Please don't memorize this list. Just know that Smoothie R does not make proteins, period. Okay? We'll talk about all these specifically. I forgot one of my prized possessions today. I have a dog liver cell under the electron microscope. And what I wanted to show you is the Smoothie R, because they gave these dogs at Abbott Labs, they gave them medicine, and they wanted to see if the Smoothie R were increasing. Now think about that a minute. If I look under a dog liver cell, I look at him before I give him the medicine, and then I look at him 2 months later, and I look at his Smoothie R, what would happen to that dog if he was making more Smoothie R? Think about that. What's happening on a cellular level?
You would need to
Remember,
some-
Smoothie R detoxifies drugs. So I give him the drug, and the body's response is to make more Smoothie R. What might happen?
resistance.
There's going to be a resistance. There's going to be tolerance. Is Abbott Labs going to want to sell a drug if they have to give you more and more of the same drug every time you take it? I would hope not. Be good for business though, right?
Mhm.
Yeah, you physically can see the difference with a drug tolerance. That should be cool. Going to be a lot cooler if we go see the Abbott Labs eventually.
So different kinds of cells have different ratios of smooth and rough ER. Like, I would
Yeah.
imagine like a pancreas cell probably has more rough ER.
They asked that question one year. I'm going to give you that question on Friday, on Participation Friday. And they're going to ask you things like that. They're going to say, what if a cell has a lot of smooth ER? Well, then I'm guessing it's probably a liver cell, right? It's detoxifying, right? Which is the job of the liver. Or it might have a lot of Golgi apparatus, which secrete the proteins, probably a pancreas, right? So yeah, you can tell a lot about a cell by what organelles it has. Questions? Nice job. And speaking of Golgi bodies, now these Golgi bodies, Gael, I don't know if I'm back to you. If you don't mind, you answer another one for me here. These Golgi bodies are going to package up those proteins. You see these little vesicles surrounding them? We're going to put the protein in there. Now think about it. We just got it all in the right shape inside the ER. I don't want it to get screwed up. This vesicle has to go out of the cell membrane. And so what I'm going to do is give it its own, very own membrane to stay intact. It's going to stay in its tertiary structure, like the pancreas producing insulin. Insulin might be inside here, right? Here, I'll put insulin inside. There it is, right? Tertiary structure. Gael, if that protein is the wrong shape, what's going to happen? Is it going to work anymore? Will it work like insulin?
It won't work like insulin since
Oh,
shape
Shape
matters.
is what matters. Yeah, excellent. Good job.
Yo, I'm
Yeah.
starting to remember.
I didn't hear
like
you
a-
again.
Yeah, like I'm starting to remember something I did in seventh grade. Because like
Yeah.
in seventh grade, we were learning about cell structures, but like,
Yeah.
I think the teacher came up with a really good way to to kind of internalize exactly what like the structures do.
Sure.
Like it kind of compared them to like a country or a city.
Yeah, that's a good one. Yeah.
Yeah, and
So you
I
guys
think
made your own city.
I think the Golgi was like Amazon or something. I don't remember.
Yeah, I like that. I like
Yeah.
that. Yeah. Golgi body as Amazon. Yeah. Packages it up, right? There should be a little arrow on there. A little logo. I like that. That's a good one. Now, if the Golgi body, if this protein's not going to leave the cell, it goes into one of these lysosomes. And lysosomes have digestive enzymes. They have hydrolytic enzymes. Again, we're not going to memorize the whole list of 50,000 enzymes in our body, But you know, one, we worked with catalase. It broke down hydrogen peroxide, right? And again, if those enzymes leave these membranes, we're in trouble because they're going to become denatured. So they stay within these lysosomes. Lysosomes are going to do things like destroy old cell parts. And two big diseases, don't worry about this top part, just this part down here. Two big diseases I'd like you to know are Pompe and Tay-Sachs. In both of these cases, these are deadly diseases because the hydrolytic enzymes that are supposed to be in those lysosomes do not occur. The DNA was wrong, so the order of amino acids were wrong, which means, as Gael just told us, the shape of the protein is wrong. Now, Pompe disease has a very famous movie after it. I want to show you this. Has anybody seen Extraordinary Measures? Later on, maybe we'll watch this movie, but I want to show you the trailer on this. Oh, I hope I'm going to show you the trailer on this.
In both kids, it's the heart that's the real threat to their lives.
How much more time do we have?
Megan, maybe a year. Patrick, less.
I wish that we had a drug to treat Pompe, but we simply don't. I'm sorry.
Dr. Stonehill. Yeah. This is John Crowley. All the researchers out there say that you're a genius on the verge of a scientific breakthrough. I'm not on the verge of anything. How much would it take to prove your theory?
The odds against you are crushing. But where does that leave your kids when the dad has no job and no health insurance?
You're right. This is crazy. I can't just sit around and wait for my kids to die. I promised them that we'd raise 500.
That's all?
1,000.
Is it 500 or 1,000?
500,000.
Are you totally insane?
Apparently.
Was he worth it? The Stonehill guy?
He's really eccentric, but his science is way ahead of everybody else's.
Do you have a wife?
Ex-wives. 2 of them.
Yeah? How come?
Because I'm so easy to get along with. Figure any dude in a business suit can help me raise venture capital and run the company. But who's going to be half as motivated as a dad who's trying to save his own kids?
We can do this. We push ourselves. We work around the clock. I already work around the clock. Great. Cure diseases in theory, but never help a single human being in reality.
I can't cure your kids, you know that, but I think I can save their lives. And what if he succeeds too late? Then what?
You're in clinical trials by the end of the year, or we pull the plug.
Nobody is going to tell me how to run my lab.
We're out of time.
Are you crazy? You've jeopardized your chances of ever getting your kids treated.
Anyway, really good movie, and it's based on a true story. I don't know how much they Hollywood it, but his kids were going to die at 8 of Pompe disease, right? Oop, I don't know what's going on with my computer right now. And again, it's the wrong protein in the lysosome, right? And the DNA is wrong. What kind of disease would this be? Because the mom and dad don't have it. Anybody remember a little bit of genetics?
The recessive.
The recessive, it's a recessive gene. So here, mom and dad got married, had no clue they had this disease until they popped out 2 children with the same disease. He quits his job and he goes try to save their lives. Really good story if you want to watch it. I would imagine you get it for free probably on, one of those shows. All right, what do I got? I got 1 minute left. I got to do this today because I already pulled it off the plant. This is it. You can put your notes away, but give me 1 more minute. You see this plant? It's in my room. It's growing like a tree. It's called cat's claw. I don't know if you can see the little claws on it. But anyway, I had a student at College of Lake County who had brain cancer. Her uncle gave her this from Mexico. This plant has proteins in its vacuoles that helps it against cancer. Now, the plant doesn't want to cure cancer, right? The plant is just producing those proteins. So if an animal like me goes and eats those, I'm eating the proteins that are in there. Just by sheer coincidence, we know that some of those proteins are helping inhibit mitotic divisions. They're helping against cancer. Her uncle swears by this plant. Now, she gave him Western medicine. They got Western medicine as well, but she was cured from her cancer, right? A lot of our medicines, about 25%, are coming from inside those vacuoles of plants. If that didn't impress you, that's the best I had today. Bye, Miss T. Have a good day.
Sorry,
All right.
I missed that. Could
See
you
ya.
say it again, please?
Now Google's talking to me. At least Google talks to me.
I seem to be having.
Bye, guys.
All right.
Thank you.
I miss it too.
Thank you. Have a good day. Sorry. Cool. Yeah, can I help get organized or anything? You need anything? Okay. I'm sorry it took so long.
Tomorrow morning.
Yep, I'll get them their bones. Yep.
They're in the freezer.
Sure.
They got a small, medium, or large in there, SML. That's just for the size of the glove.
Yep.
For them to put on
Right,
if
right.
they want to wear one.
Yep.
Just